The dendrite is the first part on the neuron. The dendrites are highly branched extensions that receive signals from other neurons. A neuron is a structural and functional unit of the nervous system. A neuron's nucleus is located within the cell body. An axon is a longer extension that transmits the signals to neurons or effector cells. The axon joins the cell body at the axon hillock, where signals that travel down the axon are generated. Many axons are enclosed in a myelin sheath. The myelin sheath insulates the axons of some neurons and helps to speed their impulses. The node of Ranvier is a small gap between each myelin sheath. Near the end of the neuron, axons divide into several branches, each of which ends in a synaptic terminal. The site of communication between the synaptic terminal and another cell is called the synapse. At most synapses, information is passed from the transmitting neuron to the receiving cell by means of chemical messengers called neurotransmitters. The resting potential is when the neuron is at rest. The outside of the cell has an excess of sodium ions, which are positive, and the inside of the cell has an excess of potassium ions, which are negative. The voltage inside the cell is at constant negative 70 MV. This potential difference is maintained by the closed ion channels and the action of the sodium-potassium pump. The dendrite depolarizes when there is a loss of negative charge in the dendrite. To reach the threshold, an external stimulus, either from a sensory receptor cells or neurotransmitters from adjacent nerve cells, causes some of the sodium channels to begin to open. Sodium ions start to move into that part of the cell, causing a local decrease in the polarity of the membrane. If this potential difference across the membrane does not reach the threshold of negative 55 MV, no action potential is generated. The neuron depolarizes through the axon if the membrane potential crosses the threshold value. Then an action potential is generated. The sodium channels open up completely, sodium ions flood into the neural axon, and the membrane is locally depolarized. This rapid diffusion causes the membrane to become oppositely polarized. That is, it overshoots the neutral polarity and reaches a potential of plus 30 MV. At this point, the outside of the cell is negative, and the inside of the cell is positive. The neuron generates electricity from these chemical events, which allow for the impulse to be traveled further and speedier. For neurotransmitters to be released, an action potential must arrive at the synaptic terminal. Molecules of the neurotransmitter are controlled within cytoplasmic vesicles in the synaptic terminal. These vesicles then bind with the wall of the synapse and open up and are released into the synaptic cleft. The released neurotransmitter molecules diffuse across the cleft and bind to complementary receptors on ion channel proteins in the receiving neuron's plasma membrane. The binding of neurotransmitters to receptors opens the ion channels. When the channels opens, ions can diffuse into or out of the receiving neuron. This triggers a new action potential, and in other cases, a neurotransmitter inhibits the generation of an action potential. The neurotransmitter is then broken down or transported back to the sending neuron for recycling or reuptake, causing the ion channels to close. This ensures that the neurotransmitter's effect on the receiving neuron is brief and precise.